Okay. Start the video. Good morning, everybody. Let's see. I'm going to share screen. Okay, we're going to view our chat. I don't really need to see my picture anymore. Okay. I'm going to go here. All right, is it still next week on Wednesday? Yes. The exam is on for next Wednesday, September 9th. Yes. Okay. I wanted to try something. Because you have two exams. I'm sorry. I'm sorry. Yeah, it's on Wednesday. A&P lab and this class. I'm sorry. Yeah, but I'm not moving it. Oh. Study early. Okay, let's see. Oh, I don't even know where I'm going. Oh, that's not what I wanted to do. Good morning. Good morning. How was your week off? How was your week off? You did a lot of studying for biochemistry, right? Yes. Yes. We'll put our attendance up, and I'm going to start the clicker cloud. Oh, no, that's not it. Oh, I'm going to the chat. What are we doing today? We are working on Chapter 3, Section 1. A&P lab and A&P class and biochemistry. Okay, let's see. Okay, we are right here. We're going to start class. Okay, moving this guy over and let's do this. Okay. So attendance is open. Y'all can go ahead and do your attendance whenever you are ready. Yes, ma'am.
Is lab schedule still the same?
I updated the lab schedule on Moodle. So it basically, we lost a week, but we were planning the week of fall break to be a makeup week. So there's no more fall. We have not received official word. I know I'm being recorded. I cannot say for sure what is going to happen with spring break. More than likely, we will lose spring break. I mean, we have to make up the time somehow. So we are anticipating that we will lose fall break and that we will use that week as a makeup week. So everything just got shifted down. You know, so it worked out that we planned it that way. Let me put my, where does my chat keep going? It keeps hiding. Where did I put it? Why can't I see my chat? Oh, there it is. Why didn't it sit on top? And Christian and Ross, if y'all need some help getting on Clicker, we need to get y'all on Clicker. Fight through it, guys. I know it's gonna be a long semester without a break, but I hope you had a really good week. Because it will be a long one. So Christian and Ross, Ross, we're gonna talk on Wednesday. So let's just talk about on Wednesday. But Christian, would you set up a Zoom meeting with me and we can figure out your Clicker situation? Okay. So if you have not done so, please do your attendance. I want my chat to sit on top. Why is it not sitting on top? There it goes. Okay, good. And Allie, same thing. Allie, Caitlin, are y'all having Clicker problems? If you're having Clicker problems, you need to set up a Zoom meeting with me and we need to figure out what's going on with your Clicker because we are far into the semester, so we need to have that figured out. Allie, Caitlin, can y'all tell me, is it Clicker problems? Like what's going on? Yes, ma'am.
Okay, so we're in A, so we don't have that updated schedule.
Y'all don't have the updated schedule? If you stay after, I'll talk about lab. Because we need to get started. How to get the link you sent to get the discount. So set up a Zoom, Allie, set up a Zoom meeting with me and I will, at the beginning of class, just not sure how to link it. So Caitlin, you should get an email that has a code and you have to set up an iCloud account on iCloud.com. If you set up a Zoom with me, Caitlin, I can walk you through it. Same thing, Allie, if you set up a Zoom with me, I can walk you through it to make sure you get the discount. But we need to get it figured out. So y'all set up a Zoom with me. On the Moodle page, how do you set up a Zoom with me? Right there, appointment calendar. Okay? All right. So please, 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 if you are just walking in, please do your attendance because it will go away shortly. Okay. So I have a few things to discuss before we get started today. Welcome back, everybody. It's nice to see everyone in the classroom. Love it. This is like got to be one of my favorite chapters. I could talk about this chapter, I don't know, until the cows come home. But so I think we're gonna have a lot of fun with this one. So before we get into chapter 3, we need to talk about things that we're supposed to be due today, which was to finish those chapter 2 discussion questions. Do you remember the discussion questions? So come down, chapter 1, chapter 2 discussion right here. So a lot of y'all did a really good job with it, but we're missing a few things. This isn't the only slide, but we are missing a few things. I will be going over it tomorrow morning, don't forget to grab a wipe, very early in the morning. So I'm only commenting if you have put something there. If you have something there and it's not right, I will give you feedback, all that kind of stuff. But if it's blank, it's blank. I'm gonna leave it, right? So if you want the feedback, I gotta have something there. Yes, ma'am. No, I'm gonna see. I see who's working on it. You can, that's fine. Because sometimes group members like need to be able to communicate. Yeah. Well, who posted what, right? So sometimes it makes it really easy. So if you'd like to, that's great. but I don't need it in order to see who's edited the document, okay? I really want my chat to come up. Okay, let's see. Okay, and Emily, Crystal, Grace, if your iClicker is not working, you need to set up a Zoom with me, and we will figure out how to fix it, but you need to set up a Zoom with me. And how to set up a Zoom, we said, was up here in my contact info right here. Okay, so if your iClicker is not working, you need to Zoom with me. All right. Okay, so you need to finish chapter 2. So I will be up at, like, I don't know, 3 in the morning tomorrow morning, and that's when I'll be checking it. So please have it done by then. Let's see, our exam is next Wednesday, September 9th, right? It will be only on chapters 1 through 3. So it just basically got shifted 1 week because we closed for a week. I really kind of think we could have done it a little bit sooner, but that's okay. I don't think y'all are going to complain. It's going to be an online exam because we can't fit everybody in the room at one time. We can't do it. There's not an open room for us. So it will be online. So it will be monitored with Respondus with a webcam. So it's a lockdown browser. You won't be able to go anywhere, and you will be videoed while you do it. You are welcome to do it here in class. You're welcome to bring your computer if it's got a webcam or iPad that's got a webcam. You can do it here in class. That's fine with me. I'm here, but what I'm going to have to do, we can't all fit. I'll set up a sign-up sheet, and I'll send out an email that says, I'll probably put it out sometime on Wednesday. I'll let y'all know on Wednesday when it's going to go out, and y'all can sign up, and the first 32 who sign up can be in this room. But then I'll also post a list of all the other places on campus that have a computer with a webcam that you can use. Okay. All right. Any questions? Oh, and I'm going to post a pretend a mock exam so that you can test run Respondus and you know how it's all going to look and all that kind of stuff. It'll be like, what's 1 + 2, right? You know, something like that, so that y'all can just get comfortable with it. Anybody used Respondus for an exam before? Oh, this should be great then. Okay, good, awesome. All right, trying to think what else. Okay, yes.
How is the exam going to be?
You'll have multiple choice and you'll have some discussion.
Like the calculations, can we have like the sheet?
Yeah, if you do it, you have to show it to the camera that it's blank. If you have 2 sheets of paper, you have to pick up each individual sheet of paper and show it to the camera and show the camera that it's blank and there's nothing else. Because there's like there's an area check where you like pick up the camera or whatever and kind of move it around so that I can see your setting. And if you're in the classroom with me, you actually do have to do the same thing, but I'll be here.
And can we use calculators?
No, you can use any calculator you want, and Respondus has a calculator. So I'll include that. And if y'all want, I'll bring some loose leaf paper on the day of the exam if you end up taking it in here. Other exam questions? And I'm not seeing my chat. I don't know why my chat keeps going away. Okay. So y'all are talking about, iCloud, Clicker. Okay. We're good on that. All right. Let's see. Oh, the other thing I wanted to point out is for chapter 3, you have the flashcards, you have the lecture slides, and I already posted the discussion questions. I usually don't post those until Friday, but I went ahead and posted them early. I thought that might help. Yeah? Okay, so they're posted. And then you have the self-check. So like you really have all the material because you have all the lecture. All we have is really clicker questions and discussion questions. And you're ready for the exam. Yeah? All right, let's see. So you should have at least watched chapter 3 through chapter 3, section 1 for today. Everybody good on that? Okay. All right, so let's get started. So I'm gonna end. Everybody done your poll for attendance? If you have not done attendance right now, everybody do your attendance because I'm about to change it. let's see. Here we go. Chapter 3. So we're going to be really careful and make sure that for each of these questions we explain why after. I think that's probably the most important thing for all of this. All right, so I am going to stop this poll and we're going to start clicker question number 1. Can I move this somewhere? Can I hide this? Hide. Okay, and I'll keep my chat. All right, here we go. Multiple choice. Oh, stop. I clicked one too many things. All right. Clicker question 1. Consider the following electron micrograph of the linear chromosomes and identify whether you are observing prokaryotic or eukaryotic DNA. Where did my chat go? Oh, stop. What's in it? I think we had 51 people check in, so I'm looking for 51 responses, but I'm cutting you off in a minute. Okay. So what you should have said, if you didn't look up at the screen, was that this is eukaryotic DNA. How do you know it's eukaryotic DNA? It's what? It's linear. It's linear, and prokaryotic would be circular. That's correct. But think about the structure of how prokaryotic DNA is organized in the nucleus. Is it condensed? Yes, it is. Yes, it is. It is condensed, but it's not condensed into this X shape, right? Eukaryotic DNA is organized into this specific shape. Prokaryotic DNA, not so much. It is condensed, because if it wasn't condensed, a bacterial cell would have to be enormous in order to fit all the DNA in. So it is condensed, but it's not condensed in this shape, and it is not linear. So that's really 2 reasons why. And before I go on to the next one, I really, really want to see if I can show my chat. Why does my chat keep going away? Okay. All right. Good. Questions on that one? All right. Here we go. Consider the images of the double-stranded DNA. In the image on the left, identify groove A in the longitudinal axis of the molecule. Is it the major groove or the minor groove? Five seconds. Okay, so what did y'all say? It is the minor groove. How did you know it was the minor groove? What's the definition of the minor groove? Like we see a small arrow and a big arrow. Okay, major and minor. But what is the arrow actually measuring? The distances between the 2 strands. What part of the 2 strands? That is correct. But what part of the 2 strands? The phosphodiester backbone. That's right, the phosphodiester backbone. So why do we need to have a minor groove and a major groove? What's the point? Functionally. Whenever you think about structure, you also need to think about function. Did y'all fall asleep over the 5 days y'all were away? Protein interactions, that's exactly right, Taylor, protein interactions. So we need places in the DNA where you can bind a lot of different proteins, like transcription factors, like polymerase, like all kinds of different things. And so a lot of times where those bind initially are in the major groove. So where there's a little bit more space for them to bind. Okay, let's try another one. For nitrogenous bases, the heterocyclic ring contains 9 atoms. Does that define all nitrogenous bases, purine nitrogenous bases, or pyrimidine nitrogenous bases? And normally I would not give you a picture with this question. Trying to ease you into it this morning. I can't see my chat today. There it goes. All right, 3 seconds. So what did y'all say? Good, good. All right. So we know that those are purines. Do you need to know the structure? Yes. Without looking at a picture, yes, you do. You do. So all purines are going to have 9 carbons inside of the ring, right? Within pyrimidines, we only have 1 ring. So if I ever say heterocyclic, what does that mean, heterocyclic? Are these cyclic? Down here, these pyrimidines? Absolutely, absolutely. Are these cyclic? Yes, but we have 2 different kinds of rings. So remember heterogeneous and homogeneous? Remember that, same and different? So heterogeneous tells us it's different, homogeneous tells us the same. So heterocyclic means that there are multiple rings and the rings are not the same. So we have a 6-carbon ring and a what? And a 5, right? Because those 2 in the middle kind of do double duty in both rings, right? For a total of 9 different carbons, okay? All right. Okay, consider the following DNA, AGGCT. If this were serving as the template strand for the new polynucleotide polymerization, how would the new strand read? If you're a biology major, you love this question. Three seconds. Okay, so what did y'all say? Should have been D. Should have been D. What's the difference between A and D? A is 5' to 3', D is 3' to 5'. So remember, what's our definition of complementary anti-parallel base pairing, right? So we know that the strands have to run opposite in the 5' to 3' direction, and the bases have to be complementary. So tell me about, is this new polynucleotide strand DNA or RNA? Can you tell? It's absolutely DNA. How can you tell? It's not uracil. You have no uracil. Okay, good. All right, let's try another one. These are kind of interesting. Within the graph of the three melting curves, where would the DNA be most likely to have sodium ions neutralizing the charge on the DNA? Will you have to interpret graphs on the exam? Yes. Okay. So what do we say? Oh, we have a big range. Let's talk about this. Let's talk about this. So we're saying either B or C. So why? Why B or C? What are we looking at? What's this a graph of? This is a graph of our melting temperature, right? And what is melting temperature? Denaturation of the DNA. That's exactly right. Denaturation of the DNA. So what happens as we increase the sodium chloride concentration? Melting temperature increases, or the temperature at which half of the DNA molecules are denatured is higher, right? So what does this tell you about how sodium chloride, now you kind of have to figure out which one is going to interact, but it kind of tells you in the question, which one's really going to interact with DNA? Tell me about DNA's charges. It's negative. Where is it negative? On the backbone. The backbone is negatives all down the sides, right? Tell me what happens when you have negative charges next to one another? They repel. They repel. So what's more stable? A basically naked DNA with no sodium, or sodium completely coating the outside, sodium ions completely coating the outside of the DNA molecule? Coated. Coated is going to be more stable. You're going to have less repulsive forces, and so it's going to be much more difficult to separate the strands. So now what do you think? B or C? C. C is going to most likely have that maximum amount of sodium ions coating the DNA. Now, will you get to a point where it doesn't have an effect anymore? Absolutely, right? You might increase it to the next, and then you'll have an overlapping curve, right? Or the distance between the curves will get smaller and smaller, absolutely. But in C, we see the most amount of sodium ions coating the backbone of the DNA. Okay. All right, so let's do one more kind of like this. The DNA sequence can affect the melting temperature. If the GC content increases, why does the melting temperature go up? It's Kayla, it's only one answer choice. Oh, I'm waiting too long. Okay, so what'd y'all say? All right, let me ask you a question. If we're talking about denaturation of DNA, We're going from double-stranded, right, to single-stranded. When the DNA is in a double-stranded, what are some of the factors that keep that double-stranded DNA together? Base stacking. That's the primary cause. What's base stacking due to? Base stacking, think bases one on top of the other. This is mostly van der Waals. Do you remember van der Waals from, like, I don't know, Chem? I don't know the numbers, y'all. I'm thinking like 101, 102, but that's not right. That's not the right numbers, but y'all know what I mean, right? Okay, so what happens when the atoms get close together, right? There's like a sweet spot. Remember this? If they're too far away, they don't attract one another. If they're too close, they actually repel each other. But there's this sweet spot of where they're attracted to one another. The majority of y'all said hydrogen bonding, but here's the problem with hydrogen bonding. What happens when I take those DNA strands and I separate them? What kind of an environment am I in? Aqueous, water. What's going to happen? How many hydrogen bonding? A bunch, right? So what's the difference in hydrogen bonding? None. So what's actually determining your melting temperature? Base stacking, van der Waals, right? And those are really, really small interactions, but over a big molecule, they contribute so much to stability, right? So lots of little interactions really contribute to keeping that DNA molecule together, right? Y'all are right, there are 3 hydrogen bonds between guanine and cysteine. There are 2 hydrogen bonds between A and T, right? That's true, but that does not explain melting temperature changes with GC content. It's really base stacking. You have more base stacking interactions with a GC pair than with AT. Okay, I put that in there so that we could, it was kind of a trick question, but Caitlin, I do like to do trick questions in class so that there can be no trick questions on the exam, right? Get those out of the way. I know it was tricky. Okay, so based on these DNA melting curves, a thermophilic microbe living at greater than 75 degrees Celsius would likely have blank GC content as compared to a similar microbe living at 37 degrees Celsius. I should have done this question first. I should have flip-flopped. That would have been probably more tricky though. Okay, 5 seconds. Okay, so what'd y'all say? Okay, so we should be higher. Good. We should absolutely be higher. But is that due to hydrogen bonding? No, it's really due to base stacking interactions. Okay. Oh, this is a good one. If a sample of double-stranded DNA is determined to be 20% guanine, what percent adenine is in the sample? My chat keeps going away. Okay, 5 seconds. Okay, what did we say? Okay, C, good. How did you figure that out? by definition, right, our percent guanine has to equal our percent cysteine. So they both have to be 20% for a total of 40. So the rest has to be A and T, and they have to be equal. So half of 60 is 30. So you have the breakdown that way. Should be able to do that. All right, now I got a trick question for you. You ready? Don't let me trick you. If a sample of double-stranded DNA is determined to be 20% guanine, what percent of this DNA sample is purine nucleotide chemistry? Okay, 5 seconds. Okay. So what should we get? We should have 50%. What did y'all say? I'm going to check. E, 50%. So really, it doesn't matter how much you have. Half of the DNA is going to be purine and half of it's going to be pyrimidine. That's our base pairing rules, right? Because for every purine on one side of the strand, you're going to have a pyrimidine. Otherwise, what if you had 2 purines or 2 pyrimidines? What would happen to the width of the DNA strand? How many rings is a purine? 2. How many rings is a pyrimidine? 1. So your width of your DNA has to be 3 rings. What would happen if I had 2 pyrimidines? It would be closer together, right? What would happen if I had 2 purines? It would be too far apart. But what we know by definition is that the backbone distance is the same, right? It's the same. It's always the same. So you always have a purine binding to a pyrimidine. So you have to have a 50-50 purine to pyrimidine ratio. That was kind of a trick question. But we're getting them out of the way, right? Does that make sense? Y'all look a little confused on that one. No? Good? All right. Okay. Single-stranded nucleic acid is expected to absorb blank UV light than double-stranded nucleic acid of similar size. More, less, or the same? Five seconds. Okay, so what did we say? It should be more. Oops. It should be more. Okay. Oh, clicking the wrong things. So what happens when you are looking at concentration of DNA? If you ever work in a lab, you're going to purify DNA at some point. I'm telling you, you will. And you have to know what concentration you have. When we get to section 3 in this chapter, it's all about different methods in biochemistry. And one of them is creating a vector or a circular piece of DNA that has an insert of interest. Well, if you're trying to do that, you need to know your concentration of your vector. You need to know your concentration of the little piece of DNA you're trying to stick in. So in order for you to do a repeatable experiment, you have to be able to find out your concentration of DNA. And we do that with ultraviolet light, right? Because we know the fluorescence intensity at a particular wavelength, in this case 260 nanometers. So if you think about it, when DNA is in the double-stranded form, the base pairs are basically locked together, right? So they are preventing absorption of light because they're so close together. What happens when we convert to single-stranded? Those bases now stick out on the side and can absorb a lot more light because there's not as much stuff blocking the light, right? So you would expect single-stranded light to absorb more. All right. And I don't know where my chat went, but I think, oh, really? Sorry, y'all. Okay. Here we go. When DNA experiences a right-handed supercoil, what is it referred to? A right-handed supercoil. I should have brought my rubber bands today. Okay, 3 seconds. Okay, what you should have gotten was negative. You should say is negative. So you can have a right-handed coil and you can have a left-handed coil. It's all about which strand of the DNA basically goes on top of the other. And negative supercoiling occurs a lot whenever you are replicating the DNA, transcribing the DNA, whatever it is. Remember when I was showing you, maybe I can do it with this, right? When I was showing you, you had a loop of DNA, and this is really a lot more like what happens in eukaryotic DNA, right? It's not a circle. We know eukaryotic DNA is not a circle, but it's attached to all kind of proteins. So it has all these little loops coming off of it. So if I have a double strand, if I had 2 strands of this, right, and I tried to pull it apart, what would happen downstream? It would start to twist on itself, right? If I had 2 strands here and you pull the 2 apart, but everything's still connected down here, what would end up happening is you would get coiling. That's negative supercoiling. So whenever you unwind 2 strands to separate them so you get single-stranded pieces of DNA, you end up inducing negative coiling. And I had to twist because I couldn't. I need to get a good model of that, but nobody really has a good one. Okay, so it happens in prokaryotes and in eukaryotes. Even though this is not a model of eukaryotic DNA, why not? It's a simple circle. But we can use it to represent eukaryotic DNA as well. Because remember, in the video I showed y'all, right, if you had a piece, you have all these proteins like chromatids and stuff like that, and you end up having loops of DNA, right? So here's a loop that can be twisted, right? Okay. Oh, but I did learn something. I learned something new. Oh, I can clear the screen down. I'm so excited. All right. Okay, consider the following data regarding double-stranded DNA. We have a positive supercoiling. We have a twist number of 50 and a ride number of 4. Calculate, oh, it's not a multiple choice. It's really a numeric. Okay. Calculate the linking number for this DNA. Oh, come on. Oh, stop it. So calculate the linking number. There is a formula. You do need to know the formula. Okay. Oh, I'll give y'all another 20 seconds. So a minute 20, I'm calling it. Okay. All right, so what did we get? Good, 54. 54 is what you should have gotten. Okay, so what's our formula? Our linking number is equal to the sum of the twist and the ri. For any given piece of DNA, your linking number is going to be the same. Because it has the same length, the same number of base pairs in it, and it has the same turns per base pair. So usually it's about 10 bases per turn, right? So all you're doing is looking at two different factors. If this is my, I don't know how well y'all can see this, but it's a loop. What's my ri in this piece of DNA, this pretend piece of DNA? What's my ri? 0, 0. How would I induce ri? I'd have to twist it somehow, right? Maybe around a histone. What's another way to induce a twist, to induce ri, excuse me, that we just talked about separating from double-stranded to single-stranded, which then induces a downstream ri. Okay, so what would happen to my linking number if I went in and transcribed a piece of DNA? So I split my double-stranded into single-stranded. I induced a downstream ri. What would happen to my linking number? So remember, it's a proportional relationship. If we separate the two, what's that doing to our twist? It's dropping our twist. So what would have to happen to the ri? It would go up. So what would my linking number do? Stay the same. But we said that linking number is a constant for a particular piece of DNA. What's the only way you can change that? Anybody? No, not even. Well, if it was a big enough deletion, yes, but it would change the number of base pairs. So yes, that would. What's a more common way? Enzymes, topoisomerases. Remember that from the video? Yeah? So topoisomerases induce either single-stranded or double-stranded breaks. If you induce breaks to relieve strain, to relieve supercoiling, what's going to happen? Are you going to change your twist? No, but are you changing your ri? Absolutely. So how can you change your linking number? You can do that with topoisomerases. Okay, let's see. Moving on, but let me clear the screen first. Maybe. All right, this one is multiple choice. Okay, the electron micrograph shows two DNA molecules that are 500 base pairs in length. Assuming 10 base pairs per turn, identify the DNA with a linking number that is not 50. And the trick is then explain why. Okay. Check in if you make a response if you have not. All right. So what did we put? It should have been A. It should have been A. So why? Well, you have to do a little calculation, right? Look at A. What's our twist? Mathematically, how do we calculate twist? Is equal to what? Our number of base pairs divided by number of base pairs per turn. Yeah, the answer is showing, yes. Yes, we're going over the answer, right? Okay. So if you calculate the twist, what do you get from twist? 500 divided by 10 is, you just cancel your zeros, right? 50. So you don't really need a calculator for that one. Tell me about rye of this one. Does the DNA ever cross over? A, it's A. Oh, it's A. Oh. Okay. Oh, no, no, no. Okay, so wait. So what's your rye of A? Zero. What's your twist? 50. What's your linking number? 50 plus 0 is your linking number. So it's 50. So A is 50, right? Now tell me about B. Yes, Ross, it's base pairs, total base pairs divided by base pairs per turn of the DNA, per helical twist. Okay. So tell me about B. We still have how many base pairs? 500, right? We still have how many base pairs per turn? 10 plus, what's our rye? Some number, but it's a number. Call it 5, doesn't matter. So cancel it, we get 50 plus 5, what do we get? 55. And it could be anything, but it's greater than 50. So your answer should have been B. What did I mark? I marked the wrong one here. Sorry. Okay, so B is correct. I'm sorry, I clicked the wrong one on cloud. So really, like, put in some pretend numbers. It helps me to understand formulas when I actually use numbers rather than like looking at a picture and going, well, I guess, right? I know that this A DNA molecule has no rye. This one has some kind of rye, so I know that that number is going to be different. My twist is the same because it's still DNA, it's still the same number of base pairs, but what's going to be different is the rye. Yeah? I feel like I a little bit blew your minds. No? I still see a few confused faces. Okay, so B is correct. So the other thing that I have here, if I would stop moving the mouse, would be really nice, right? Right, so A, how would we describe A? We would say B is supercoiled, right? It's overwound and it's supercoiled. You really can't tell that just by looking at the picture, but you can tell it's supercoiled. What would you call A? There's a term for that. Relaxed. It has no rye, it is relaxed. So B could, if you were just looking at the picture, you weren't given any numbers, anything like that, could be either, could be is supercoiled, but could be overwound or underwound. And that tells you if it's a positive or a negative twist. Okay. All right. Oh, gotta clear my ink. All right, I think this is like one of my last ones from this. That works out pretty well, right? Let's do this last one. This is kind of cool. Cancer therapeutics are designed to impact the life of the replicative cell in some way. Some chemotherapeutic drugs target topoisomerase enzymes. Which of the following statements most accurately describes the effect of these types of anti-cancer drugs? And I'll give you all a minute and a half because it's a lot of reading on this one. Okay, 5 seconds. Answer if you haven't answered. Okay. All right, so what do we say? Should be A, good, should be A. Okay, so they negatively impact DNA replication. Why do they negatively impact DNA replication? If it targets topoisomerase can't work. So when do you need topoisomerase? When you are replicating, when you are transcribing, but big on, it's more impactful on replication in cancer cells. Why? They're what? But don't regular cells undergo? Well, it's not a receptor. It's the binding. It would inhibit the binding. That's right. But would it happen in a regular cell? Why not? We're working on drugs now that are like conjugates, where you take a little piece of DNA, RNA, protein, and you attach it to your cancer therapeutic, so that that little tag is only recognized by cancer cells, and then only cancer cells pick up the chemotherapy, pick up this anti-topoisomerase, whatever. But that's not on the market yet. That's in research. So are you affecting normal cells as well as cancer cells? Absolutely. Why does it say, though, that we're targeting cancer cells? What's a characteristic of a cancer cell? Uncontrolled growth over all the time, right? So anytime you have a drug, you can basically pick on that characteristic, right? So will it affect normal cells? Yes. It is more dangerous for cancer cells because they have to replicate so often. So if you inhibit any part of the replication process, it is going to be more detrimental to cancer cells, because in order for cancer to survive, it has to keep replicating over and over and over again. I mean, that's so you're gonna, by inhibiting topoisomerase, you're gonna inhibit the ability to relieve the supercoiling that occurs when you have to separate the strands. So replication is not going to go. But are you going to have an effect on other cells? Yes. I mean, you can inject a tumor, yes, but you are going to have some off-target effects. This is one of the things that I think you can write this down and see if I'm right in, I don't know, 15, 20 years. Y'all, but I think this is the direction that we have to go in order to make cancer therapeutics actually work, is to design a delivery system where you are delivering to specific cancer cells, not just anybody's cancer cells. Like, you would have to take a sample of the patient's cancerous cells out of their body, grow them in tissue culture, then design some kind of a delivery system that is only recognized by their cancer cells and not recognized by their normal cells. Because every single type of cancer is unique. It's unique from person to person, even if they have the same leukemia, right? Everyone is unique. So until we design drug therapeutics that are unique, you're going to get off-target effects. That's what chemo is. Chemo's horrific. I hope that somebody stands up here in a classroom in 50 years and says, can you believe what we used to do to treat cancer? We used to give them things that all cells would take up, but the cancer cells would take it up more, and so they would die. But what happens to a patient who's undergoing chemotherapy? What are their symptoms? They're sick, they're nauseous. What else? Weight loss, hair loss. So they don't eat because they're sick. But why are they sick and why do they lose their hair? But why those two particular symptoms? What grows very rapidly? Your hair and the cells that line your stomach that create mucus. So if you kill the cells that line the stomach that create the mucus, there's no mucus to prevent the stomach acid from basically eating itself. And then you feel sick, right? So we need y'all to go out, become researchers, and figure out how to do that. All right, that's your homework. Y'all have a good rest of your day. How long did I talk? Ooh, I gave y'all 2 minutes. If you have iClicker questions and you're in the class, you can come up and I can help you. But otherwise, if you're on Zoom, you can either set up a Zoom with me or you can stay after class tomorrow and I'll help you then. Either way is fine. So everybody on the chat okay? Anybody got any questions? Okay, so I will end the Zoom if everybody is good. I'll give y'all another 30 seconds.
I have a question.
If y'all need anything, and I'm going to end.
Sure.
I'm Madison. This is my first day back after COVID.
Welcome back.
So my class is like right next door.
Yeah.
Is it okay
If you wait and let everybody out, like just sit for a minute and wait and let everybody out, then you're good. You can go down. I mean, it's good. We just don't want people crossing over. So just let them go out and then y'all can do that. Yeah. Okay. So I'm going to end because I have a question. Okay, Cecilia, what you got? I'll try not to end it. What you got?
My iPhone for some reason wasn't working today.
Okay.
But I wrote down the answers.
So cool.
I don't know if it counted my attendance or not.
Oh, let's check.
I think it said I had my attendance and then it just was like closing me out.
Oh, that's weird.
So I'm gonna go work on that once I'm home.
Okay, wait, let's see. Let's do it while it's still open and let's see what happens. Let's try attendance first. And
I had bought it already.
it says
So it doesn't check in.
I don't see it right now because it's currently going. Wait, let me refresh this. Okay. Me and another biochem student live together. How are we supposed to take the exam? What do you mean? You might have to go to different locations. There are different locations. I'll post them on Moodle of locations on campus where you can use the computers, and the computers have a webcam, or one could come to the classroom.
I don't think my microphone is on.
So checked in. Let me see. Who's checked in? Annie's checked in.
Okay. I'm gonna just figure out what it's doing. So
Okay.
my phone's been having issues of late anyway. So
Well, you can always do it on your laptop.
you can do it on your laptop?
Yeah.
Oh,
Yes, you can do it on your laptop. Try that next time.
I will do that. I'll
Try it on your
download the
laptop
app
next time.
and then for next class, come and try it.
Yes. Okay,
Thank
Cecilia,
you.
does that answer your question? I could actually turn the audio on if y'all want to talk. Yes, it answers your question. Okay. Anybody else have a question? Let's see. My volume's up if anybody's got a question. I don't see anybody else. Anybody? Questions? All right, I'm going to end. I will see everyone tomorrow. Not tomorrow. Wednesday.